Hello fungus and welcome to the first channel about fungi. Today we will talk about Conidiophores and Conidia.
Conidiophore is a structure of the asexual reproduction of fungi. It bears asexual spores, Conidia.
The asexual reproductive cycle can be divided into two main phases: phase of vegetative growth and phase of development.
Vegetative growth phase starts from the germination of Conidia, creating germlings. Germlings is a Conidia with young hyphae, like you can see on this illustration. Next stage is a sustained filamentous growth, or hyphal growth and development. And then hyphae interconnect with each other and form a cell network called mycelium.
After a certain period of vegetative growth, which is individual for specific fungal species, some hyphal cells stop normal growth and begin asexual development,
which includes conidiophore formation and maturation of asexual spores, Conidia. Conidiophore formation starts from thick-walled hyphal cells, food cells, which extend into the air, creating aerial hyphae.
And these extended hyphae are known as stalks.
Tip of stalk swells and forms a multinucleate vesicle. Vesicle gives rise to two layers of uninucleate reproductive cells, the supportive cells medulla and phyalides. By the way, each medulla produces two phyalides. Phyalides are subjected to repeated mitotic division, creating chains of conidia with a single nucleus. The youngest, immature conidium adjoins the phyalides, and matured conidia are located towards the end of the chain.
Conidium maturation is a differentiation process occurring after the formation of the septum between the phyalides and the newly formed conidia. As a result, matured conidia detach from the phyalides and spread around.
The final multicellular conidium-bearing structure is called conidiophore.
And I think it's very exciting that only one conidiophore can produce more than 10,000 conidia.
Conidiophore that has both rows of cells, medulla and phyalides, is termed biseriate. Biseriate means two rows of cells. For example, Aspergillus flavus has this kind of conidiophore. If conidiophore contains only phyalides connected directly to the vesicle without supportive cells medulla, this kind of conidiophore is called uniseriate, which means one layer of the cells. An example is Aspergillus fumigatus. Regarding the conidia, there are two possible scenarios. In the absence of nutrients, mature conidia enter an extended resting state, a state of dormancy.
In addition, in this state, conidia can overcome different harsh environmental conditions, like for example extreme temperatures or high osmolarity, until the environmental conditions will be appropriate for germination.
If the environment is conducive to fungal development, conidia initiate germination and fungus transits to the phase of vegetative growth.
The entire process of vegetative growth, conidiophore formation, and maturation of conidia is genetically determined for specific fungal species.